I'm today to talk to you about the our NSAIDs project, where we use Benchtop 19F NMR spectroscopy to enable mechanistic analysis and catalytic optimization for the preparation of Celecoxib and Mavacoxib, the aforementioned NSAIDs. All right. So some initial background. Fluorinated compounds make up a wide subcategory within the larger library of medicinal compounds. Of these, 18 have been classified as FDA-approved non-steroidal anti-inflammatory drug compounds, or NSAIDs, NSAIDs. Our study in particular focuses on two of these organofluorides, CoCib2 inhibitors, Celecoxib and Mavacoxib. Both of these compounds are synthesized by the same postulated mechanism, a pyrazolyl cyclo-condensation of a substituted hydrazine and diketone, as shown in the figure to the right. And the only distinction between the two of them would be the functional group on the pair position of the aryl diketone, a methyl for Celecoxib and a fluoride for Mavacoxib. Through the use of 19F NMR spectroscopy, our project aims to elucidate a deeper understanding of this reaction mechanism by analyzing NMR samples gathered around the clock. Okay. In each trial, we observe the time course for the conversion of our starting materials into product, looking for key, potentially rate-determining intermediates, and comparing the efficacy of different solvent catalyst conditions from a selection of both protic and aprotic solvents, as well as Lewis and Brønsted acid catalysts. Over a couple months, we would gather the data pertaining to upwards of like 1,000 NMR samples, from which we were able to discern two interesting details. The first is that, as shown through the time course to the right here, there seems to be no slow step intermediate in the postulated mechanism, with the rate determining step being the initial attack of the primary amine on the 3-carbon of the diketone. However, the second detail was the inconsistent nature of some of the kinetics trials and its relation to a new possible variable, the rate of keto-enol tautomerization. Okay, so to the left is a representative example of many of our more irregular samples. In the blue and green are the concentrations of keto and enol diketone tautomers, and in the red, the concentration of the product. What we observed in these trials was that the slow rate of the keto-enol tautomerization led to the concentration of product increasing at an abnormal pace, as you can see through this kind of ugly like red line coming up right here. And in contrast, we have equally numerous triplicates where this process happened at a much quicker speed, and the results were much more reliable, having a low error rate and forming a smooth first-order curve as the reactants were used up and the product concentration increased over time. All right. With this in mind, a new question of interest became clear. How does the equilibrium constant of the tautomerization of our diketone starting material affect the rate of pyrazolyl cyclo-condensation, along with other metrics such as formation of a regioisomer? Through DFT analysis, we concluded that the enol tautomer was more favorable than the keto one by over 5 kcals per mole, further prompting us to pursue a number of different comparisons pertaining to the varying solvent-catalyst equilibria and the subsequent rate of the reaction. Some factors we hypothesized could also correlate to these different variables include the solvent's dipole moments and electral permittivity, and perhaps even the atomic radius of our different metal triplet acid catalysts. So in light of these new hypotheses, we have recently begun running a new set of series of experiments. We closely monitored the process of keto-enol tautomerization in the same library of solvents, paired initially with scandium triflate and tosic acid, which are just examples of the aforementioned Lewis and Brønsted acids, and determined their equilibrium constants as well as the time necessary for the ratio to be established. As it turns out, it seems that ethanol and methanol, our chosen polar protic solvents, consistently exhibit decent amounts of keto tautomer at equilibrium, shown through these graphs. Meanwhile, DMF, DMSO, and NMP, our polar aprotic solvents, seem to generate negligible amounts without the aid of triplet acid catalysts. So in the last month, we have gathered the data points necessary in comparing all for one of these solvent conditions and all in these catalysts, and we compare the keto-enol ratio and initial rate of reaction in ethanol, which has yielded consistent results and a promising trend, as shown through the 0.99 R-squared value of the current trend line, thus far aligning with our new hypothesis, something we hope stays true as we continue our experiments. All right, so just some acknowledgements. Thank you very much, of course, to all of my group members who have been doing a lot of this work. I mean, all these time courses take like 6 hours, maybe more, and we've been through a lot of stuff for the past few months. Special thanks to especially like Andrew. Andrew's been really good at like getting this stuff done real quickly, really good on the analysis, and of course, thank you to Ed for providing us with this opportunity and providing us with guidance throughout this project. Thank you.